Everyone, welcome to Our Lady of Fatima College of Medicine. I am Dr. Pedro Ampil from the Department of Anatomy. I am a graduate of Forestern University, Nicanor Reyes Medical Foundation College of Medicine. I am a fellow of the Philippine Society of Anatomists. Now for today, we will take up two topics, namely cells, structures and functions, and cell parenchyma and stroma, okay, together with the cell characteristics. Now, our reference book in histology is Junqueras' Basic Histology, the latest edition in the lecture, and in the lab, our reference book is De Furis Atlas of Histology. Now, before we go into our lecture, let us first cite our objectives. Now, at the end of the semester, students should be able to first identify the main or basic parts of the cells. Second, enumerate the structures, components, and functions of the cell membrane. Third, discuss the different organelles, inclusions, and cytoskeletal components of the cytoplasm. Fourth, understand the role of the nucleus and its component structures. Fifth, describe cell division and its stages. Sixth, discuss some clinical conditions associated with the structure and functions of the cells. And seventh, define parenchyma and stroma and cite examples. Okay, so let us now go into definition of terms. First, let us define histology. Now, histology simply means the study of tissues. Now, tissues are groups of cells similar in structure, function, and intercellular substances. Now, cells, these are the smallest structural and functional units of the body. And the study of cells is called cytology. So we study cells with the use of microscopes, okay? The light and electron microscope and other forms of microscopy. Okay. So let us now go into the basic parts or principal parts of a cell, namely the cell membrane, also known as plasma membrane or plasmalemma. Next, the nucleus, and then the cytoplasm. Okay. Now this is an electron micrograph of a cell, okay, showing its basic parts, the cell membrane here, okay, and then this is the cytoplasm, and this is the nucleus, okay. So these are the basic parts or main parts of a cell. Okay, so now let's go structure of the cell membrane, okay, under electron microscopy. Okay, now the cell membrane under electron microscopy has three layers, okay, namely you have an outer, okay, and an inner phospholipid layer and a middle lipid bilayer, okay. So the cell membrane has a trilaminar structure under the electron microscope. Now regarding the molecular structure of the cell membrane, okay, here you have, okay, the phospholipids, okay, bilayer. Now this phospholipid bilayer, they contain polar heads, okay, which are hydrophilic, and non-polar tails, which are hydrophobic. Now when I say hydrophilic, it means that they have an affinity for water, so that these polar heads, they are directed outwards towards your extracellular fluid, okay? Now, and when you say hydrophobic, it means that they don't have any affinity for water, so that these non-polar tails, they are directed towards the middle of the cell membrane, away from the extracellular fluid. Okay. Now, this middle lipid bilayer has a fluid consistency, okay? And as a result, okay, the compositional structure of the cell membrane is characterized as a fluid mosaic model. Okay. So now let's go into the composition of the cell membrane. Now the cell membrane is made up principally of the following. First, you have carbohydrates here in the form of oligosaccharides and glycolipids. Okay? And then we have fats in the form of phospholipids and cholesterol. Now cholesterol, okay, is involved with the fluidity of the cell membrane and likewise the movements of the membrane. Okay? And then we have proteins here. Okay? In the cell membrane, now proteins make up more than 50% of the cell membrane weight. Okay? Now there are two types of proteins. Here we have your integral proteins. Okay? And here we have your peripheral proteins. Now the integral proteins class, these are directly incorporated within the middle lipid bilayer. Okay? While the peripheral proteins, they are loosely associated with the inner and outer surfaces of the cell membrane. Now, an example of your integral proteins would be these transmembrane proteins here. Okay? Now, these transmembrane proteins, they span or extend across the cell membrane. Okay? Involved with the transport of materials and acting as channel receptors. Okay. So we're through with the structure and composition of the cell membrane. Okay? Now let's go into, okay, the transport mechanisms that occur across the cell membrane. Now here you have two processes, namely your passive process and active process. Now in passive process class or in passive processes, okay, there is the movement of substances from a region of a higher concentration into a region of a lower concentration without the expenditure of energy. Okay? Now examples of this, we have your simple diffusion, osmosis, facilitated diffusion, and filtration. Okay. Now we go into the active processes. Okay, here, okay, again, you have movements of substances across a concentration gradient with expenditure of energy. Okay, and the energy source is hydrolysis of your adenosine triphosphate. Now, examples of this you have, okay, active transport mechanism involving your sodium pump mechanism. And then we have exocytosis, okay, which involves the uptake of materials enclosed in a membranous vesicle from the interior of the cell to the outside or exterior of the cell. Okay? And then we have endocytosis. Okay, this involves folding and fusion of the cell membrane, okay, producing vesicles that enclose the material to be transported from the exterior or outside of the cell into the interior of the cell. Okay? Now, examples of endocytosis, we have the following. You have phagocytosis, pinocytosis, and receptor-mediated endocytosis. Now, in phagocytosis, okay, this literally means cell engulfing or cell eating. In other words, the cell membrane forms drop-like invaginations around foreign bodies or foreign particles, and then they would engulf or eat these foreign bodies, forming, okay, vessel or vesicle-limited membranes known as phagosomes, okay, which are then ingested by the lysosomes. Next, we have pinocytosis, which literally means cell drinking. Okay? In other words, the cell membrane again forms drop-like invaginations around fluid particles. Okay? And they would drink, okay, these fluid particles, forming pinocytic vesicles. Okay. And then we have this type, receptor-mediated endocytosis. Now, in this process, specific molecules in the extracellular fluid bind to receptors in the cell membrane, okay, and are then taken into the interior of the cells. Okay, so those are the different things occurring across the cell membrane. Now we go into, okay, these modifications or specializations of the cell membrane. Okay, namely you have this, the microvilli, the cilia, okay, and flagella. Now, microvilli, these are non-motile structures that increase the absorptive surface areas of the cells, okay? The examples of microvilli, we have this, striated borders, brass borders, and stereocilia. Now, striated borders, these are found in cells lining the gastrointestinal tract, specifically the small intestines. Next, we have brass borders. Now, cells provided with brass borders can be found in the cells lining the proximal convoluted tubules of the kidneys. And then we have the stereocilia. Now, this can be found in some cells lining some testicular ducts, for example, the ductus epididymis and the ductus efferens. Next, another specialization are the cilia. Now, cilia are motile structures, okay, that facilitate the movements of fluids and substances across the cell membrane. Okay? Now, ciliated cells can be found in the cells lining your upper respiratory tract, okay, and in the cells lining the accessory female reproductive organs. Okay? Now, the flagella, also, these are motile structures, okay, and are seen in your sperm cells or spermatozoa, and they are responsible for the motility of the sperm cells. Okay. Now, as to the functions of the cell membrane, okay, they give the shape to the cells. Also, they separate the cell from the neighboring cells. Also, they exhibit selective permeability, okay, allowing only certain substances to pass through. Okay, and they also act as antigenic determinants, serving as recognition sites, okay, that render the cell surface its immunochemical properties. Okay, with the cell membrane. Now we're going to the nucleus. Okay, the nucleus class, it is the control center of the cell, also the command center of the cell, containing the chromosomes, okay, consisting of genes which are the bearers of our hereditary traits. Okay, the nucleus is also the site of replication of DNA and transcription of RNA. Now, under the hematoxylin eosin stain, the nucleus is basophilic, meaning it is bluish or violet in color because of nucleic acids. Okay, now let's go into the parts of the nucleus under electron microscopy. Now, this is the nuclear envelope or nuclear membrane. Now, the nuclear membrane or envelope, again, under electron microscopy, it has two layers, namely an outer nuclear layer and an inner nuclear layer. And in between, there is a space, the perinuclear space. Now, the outer nuclear layer contains attached ribosomes, okay? And they are continuous with your wrap surface endoplasmic reticulum. Now, the inner nuclear layer, on the other hand, contains proteins, okay, known as laminins, that help stabilize the nuclear envelope. Now, at some points along your nuclear envelope, the outer nuclear layer fuses with the inner nuclear layer, forming openings known as nuclear pores. It is through the nuclear pores where substances from the nucleus go into the cytoplasm, or from the cytoplasm, substances go to the nucleus. Okay, next, we have your chromatin materials. Okay, now there are two types of chromatin materials. Okay, you have your euchromatin materials and the heterochromatin materials. Now, the euchromatin materials form the dispersed part of the chromosomes, okay, forming these light staining areas over here, okay. Now, the euchromatin materials, they are metabolically active, okay. Now, the heterochromatin materials, they form the condensed part of the nucleus, okay, and they form these dark staining areas in the nucleus, okay. Now, the heterochromatin materials, they are non-metabolically active. Okay, next we have here the nucleolus. Now, note the structure of the nucleolus. they have a fibrous portion or pars fibrosa, a granular portion known as pars granulosa, and an amorphous portion known as the pars amorpha. Okay? Now, the nucleolus is strongly basophilic because of its function. Okay? It's involved with the synthesis of ribosomal RNA. Okay? And then we have your nucleoplasm. Okay? Now, the nucleoplasm is a gel-like matrix containing your chromatin materials. Okay. So we're through with the nucleus. Now we go into the cytoplasm. Now, the cytoplasm, okay, it is semi-fluid, okay, and is colloidal in nature, okay. Now, the cytoplasm contains organelles, okay, inclusions, and the cytoskeleton or the supporting framework of the cells, okay. Now, note that under your hematoxylinucin stains, the cytoplasm is acidophilic, okay, meaning it is reddish or pinkish in color. Now, this is due to the presence of proteins here in the cytoplasm, okay. So, let us now go into the organelles, okay. Now, organelles are living substances in the cytoplasm performing definite or specific functions. Okay, let's cite first these different membrane-bound organelles. Okay, so we have here an electron micrograph show the structure of your mitochondrium. Okay, note that it has two layers, an outer membrane layer and an inner membrane layer. And then you have a space, the intermembrane space. Note that the inner membrane layer class, it is thrown into infoldings called cristae that increase the surface area of the mitochondrium. And within the mitochondrium, you have your mitochondrial matrix containing DNA, RNA, and mitochondrial enzymes. Okay? Now, the mitochondrium is the source of energy of the cells. Okay? And sometimes it's known as the powerhouse of the cells. Okay. Next, the endoplasmic reticulum. Now, under the electron microscope, the endoplasmic reticulum consists of paired membranes, okay, sacules, vesicles, and tubules with flattened cisternae. Okay? Now, there are two types of endoplasmic reticulum, the rough surface and the smooth surface. Now, this is your rough surface endoplasmic reticulum. Okay, note that it contains attached ribosomes. Okay, so the function of the rough surface endoplasmic reticulum, due to the presence of these attached ribosomes, they are involved with protein synthesis. Okay, now let's go into the smooth surface endoplasmic reticulum. They do not contain attached ribosomes. Okay, now as to their functions, okay, smooth surface endoplasmic reticulum, they are involved with steroid synthesis, also involved with glycogen and cholesterol metabolism. Also, they are involved with detoxification of toxic substances, okay, like drugs, alcohol, and insoluble materials, okay, produced by the body, okay. And also the smooth surface endoplasmic reticulum, okay, it is involved with the release and recapture of calcium ions for the contraction of skeletal muscles. Okay, the Golgi bodies, complexes, or apparatus. Now again, under electron microscopy, the Golgi bodies consist of sacules, vesicles, and vacuoles, okay, arranged in parallel stacks like this, okay, arranged in parallel stacks, okay, containing a cis phase or entrance phase, okay, or a convex surface, and a trans phase or exit phase or a concave phase, okay. Now, the Golgi bodies or complexes, they are involved with the accumulation and concentration of the secretory products of the cells, okay. In other words, They are the packaging areas of cells. Next, we have here the lysosomes. Again, under electron microscopy, appears as dense spherical structures, okay, containing hydrolases or hydrolytic enzymes, okay. Now, these are involved with intracellular digestion, okay. Now, there are two types of lysosomes. You have your primary lysosomes containing inactive enzymes and secondary lysosomes containing active enzymes, okay. And then we have your peroxisomes here, also known as microbodies. Now, they are structurally similar to lysosomes, although they are smaller in size. Okay? Now, peroxisomes or microbodies, they contain also enzymes called oxidases and catalases. Now, the oxidases, they oxidize substances into hydrogen peroxide. Okay? Now, this hydrogen peroxide, it is toxic to the cells, okay? So that the catalases, okay, would break down this hydrogen peroxide into oxygen and water, okay? So those are the membrane-bound organelles. Now, we're going to the non-membrane-bound organelles, okay? We have your ribosomes, okay? Now, the ribosomes, again, these are involved with protein synthesis, okay? Now, there are two types of ribosomes, the free ribosomes and the attached ribosomes. The free ribosomes, they synthesize proteins for self-utilization, while the attached ribosomes synthesize proteins to be secreted by the cells. And then we have the proteasomes, okay? Now, these proteasomes, they are cylindrical bodies, okay, which are involved with removal of denatured proteins or proteins that are not needed by the cells. Okay? So those are the different organelles. Now, regarding the inclusions, these are inert or non-living substances in the cytoplasm that do not perform definite functions. Okay? Examples of inclusions, you have your glycogen, lipids, vacuoles, crystals, pigment granules. Okay. So now we go into the cytoskeleton. Okay, the cytoskeletal, okay, framework or structures of your cell. Okay, they form the supporting framework of the cell, also involved with cell shape and cell movement. Okay, now there are three types of cytoskeleton, namely the microfilament, exemplified by your actin filament. Okay, then we have the intermediate filaments. Okay, and then we have your microtubules. Okay, now the microfilaments class, these are the thinnest of the cytoskeletal component. Okay, and our example, of course, is the actin filament. Now, the actin filament, okay, this is distributed throughout the cells as anchors occurring at cell junctions. Also, they form the core of microvilli, and it interacts with myosin, okay, for the contraction of your skeletal muscles. Next, we have your intermediate filaments, okay. Now, this is involved also with cell shape, okay, and cell movement. The examples of this includes keratin, okay, found in epithelial cells, vimentin found in mesenchymal cells, desmin found in smooth and striated muscles, okay, neurofilaments found in nerve cells or neurons, glial filaments found in neuroglial cells, and lamin filaments found in the nuclear envelope. And then we have the microtubules, the thickest of the cytoskeletal components, okay. Now, the microtubules also is involved with cell shape and serving as tracks for movements of organelles, okay, with alpha and beta tubulins. Now, these microtubules originate from centrosomes, okay, which consist of a pair of centrioles here, okay. Now, we go into examples of microtubules, okay. First would be the centrioles here, okay. Now, again, under electron microscopy, okay, the centrioles appear as a pair of cylinders, okay, containing 9 microtubule triplets arranged in a pinwheel array, okay, again, containing 9 microtubule triplets arranged in a pinwheel or circular array. Okay? Now, as the functions of the centrioles, they determine the polarity of cells. Okay? Also, they induce formation of basal bodies and microtubules in the cilia and flagella. Okay. Next, the cilia. Okay? Now, under electron microscopy, okay, the cilia consist of microtubular cores of 9 peripheral doublets, okay, surrounding 2 central microtubules. Okay? Now, the cilia class, regarding its motility, okay, the motility of the cilia is due to motor proteins dynein, okay? This, dynein arms, okay, this is responsible for the motility of the cilia, okay? Which this exhibits ATPase activities, so that it uses the energy of ATP hydrolysis for the movements or beatings of the cilia. Okay? Now again, ciliated cells can be found in the cells lining your upper respiratory tract and in the cells lining the female secondary reproductive organs. And then we have, okay, the flagellum or flagella with the same structure as that of the cilia. They also contain 9 microtubule doublets, okay, surrounding 2 central microtubules, okay, also with dynein arms. Okay, now flagella, okay, these are seen in sperm cells and are responsible for the motility of the sperm cells. And then we have the basal bodies, okay. Now these basal bodies, these are structurally similar to the centrioles with 9 microtubule triplets in a circular array. Now the basal bodies control the assembly of axonems at the bases of the cilia and flagella. Okay, so we're through the structure and functions of your, okay, cells. Okay, now we go into your cell division. Okay, we have 2 cell divisions, mitosis and meiosis. Now we will discuss more of this when we go into embryology, when we discuss gametogenesis or the formation of the male and female gametes. Okay, so we will discuss more of your mitosis and meiosis during gametogenesis in embryology. But basically, mitosis, okay, mitosis or mitotic cell division, it occurs among somatic cells, okay, composed of 4 stages, the prophase, metaphase, anaphase, and telophase stages. Okay, the resulting, okay, or the result would be the formation of 2 daughter cells, okay, which are diploid in number, meaning they have 46 chromosomes, and they are genetically similar to one another. Okay? Now, how about meiosis? Now, meiosis class, okay, it is undergone by germ cells or gametes, okay, meaning undergone by your ova or ovum in the females and the sperm cells or spermatozoa in the males, okay? Now, there are 2 cell divisions here, or 2 maturation divisions. You have meiosis 1 and meiosis 2. Okay? Now, the result will be the formation of 4 daughter cells, which are haploid in number, meaning only 23 chromosomes, okay, containing only 23 chromosomes, haploid in number. And, okay, these 4 daughter cells, they are genetically different or unique from one another. Okay, so that concludes our, okay, discussion of the cells, okay, structure and functions, and cell divisions.